connected. Hello, good morning to everyone. Can you hear us?
Yes.
Okay. So it is my pleasure today to introduce you Dr. Nico Mertens, who is an expert in biotechnology research and industry. So Nico started his experience as a PhD and then postdoc at University of Ghent in molecular biology, recombinant protein expression, and molecular immunology. Then he got an MBA from the Management School of Brussels, and then he joined the Flanders VIB Institute, Inter-University Institute for Biotechnology, where he was asked to lead the molecular immunobiotechnology unit. And he managed a facility there, protein service facility. Then he became a director antibody engineering company based in Portugal and UK. And then Nico has collected a very long experience through these different experiences in company and universities, and now he works as an independent consulting with several companies, and is also project manager, and in particular his expertise is in lead identification, optimization and development. And last but not least, his surname is reminds us about our loved football player, so I trust him as a goal maker. Thank you, Nico.
Thank you, Claudia, for the kind introduction. So, yeah, Dries is my brother, actually. So, just joking. I'm much bigger than him. You have another great Belgian football player at the moment. He's called Kevin De Bruyne, and you'll see he's one of the best players in the world, and he's gonna do a lot for your team there. Okay, great. So yeah, my specialty is both in recombinant proteins, recombinant protein engineering, modeling, etc., and then especially in antibody engineering. And I'm working on several projects at the moment. So Claudia said I was working as a consultant. That means okay. I'll speak up. Uh, Meaning that I'm involved with several companies. So one of my roles is to create companies and to guide them in order to go for their first financing round, and then in the end going completely for a Series A financing. So that not in decades or years. So we do that. Not me alone, but we are with the team, and we do that in a time span of 2 years. So it has to be done in 2 years. They have to have proof of principle. They have to have seed foundation, and then we go to Series A in a really short time. So that's why they hire the experts. Today, I'm not going to talk about that work because that's confidential, but I'm going to try and give you kind of like an overview as where we are with bispecific antibodies or multi-specific antibodies in general, and what are the directions that will probably be emerging in the coming years. So in order to do that, let's start with looking at monoclonal antibodies, because bispecifics is just like they are derived from the monoclonal antibodies. So where are we now with monoclonal antibodies? I don't think you need to be convinced. They are a main part of our therapeutic arsenal at the moment. And this slide gives you an overview of the top 20 of monoclonal antibodies. And they're nice, they all have their logos, etc. But let's look what that really means. So this table gives you the same 20 that that we discussed there, but then with their application, the sponsor there. But what is particularly important is I put there in the last 3 columns. Yeah, so over here you see the revenues in 22 versus 23. As I need to update the slide because now we know also the revenues of 24, but it will be in the same line. And then the percentage of change. So that's interesting. So most of them, you see, the revenues are still growing. But one of the things I want to point you out is like here, the total of revenues for the top 20, only for the top 20 of these antibodies, is $140 billion. That's huge. That's not small business anymore. That is huge. Another thing I want to point you out is that most of them, the most of the remember, these are the 20 top, so these are the champions. There are more than 600 antibodies over there in the clinic. So the main part of them, they go between 3 and 5 billion dollars revenues a year, right? And then we see that there is one over here, the pembrolizumab. On its own, it has a revenue of 25 billion. I may be boring you with these economic facts, but I want to compare that with the bispecifics also. Another thing I want to point out, 2 more things I want to point out. One is the anti-TNF antibody, which for years has been the champion of all the antibodies, and you see that now is market share is going down. So there is a top always. So you get an increase in revenues, but then you have competitors, and we all know that also for the anti-TNF there's a lot of competitors. The antibody is off patent, it's not protected anymore, so then you get a decrease in the sale of the primary product, and then the other products go up. So that is a good thing because it gives you the opportunity for competitors to also have a little bit. So they will never go to 20 billion anymore, but if you can help with your company or whatever, and you can help to get a product on the market which has a revenue of 1 billion, I think that is also a nice thing to believe in. The last thing I want to point out here on this slide is here, the one in blue, and that is emicizumab, and that is the first bispecific actually that we encounter here today, and it is here in this table of the 20 best-selling antibodies. So because it's also an antibody, it's a bispecific antibody, we'll talk about it in a while, but it is there. It's there with its foot in the door. It has its place in therapy. It wasn't easy. So emicizumab was one of the first, and this is how it looked like. It's a complete antibody. It has an Fc, so they had to make it bispecific, meaning that it had to heterodimerize, and they had to find downstream processing, so purification techniques, in order to really purify the bispecific and not combinations of the monospecific. So an antibody has 2 Fcs. It has to be bispecific, but it also has 2 light chains, so the light chains were also. So this was a technology that they used, where they used the common light chain. Can you hear me? Yeah. So the common light chain, so they use libraries in order to find binders that bind A and B with the same light chain, because otherwise it was too difficult. Now I highlighted here, so this was developed in Roche with the people from Genentech. In my opinion, these are the experts in antibody engineering. They're the best, and it took them 10 years in order to develop this molecule and to be able to get that on the market. But it works, and it's a very good example. Now, we're still at antibodies, but in the meantime, we had a lot of expertise in how to engineer antibodies, and this goes for normal antibodies, but also for bispecific antibodies the same. So what we can do, obviously there's the binders. You have to find good binders, but once you get good binders, you have the Fc portion. The Fc portion is just an Fc portion. No, it's not. So you can do a lot with that FC portion. You can choose the class like IgG1, IgG4, whatever, but you can engineer the regions where the FC portion interacts with the FC gamma receptors, and that can enhance the ADCC or phagocytosis functions, or it can knock them out, whatever you want. Sometimes you don't want these functions, so you can make a silent FC, or you can make an ADCC enhanced FC. Also the latter, also by glycoengineering, which is another way of doing it, but there are ways of engineering the function of this FC. So why would you knock it out? Well, if you want to target a receptor, for example, and you want to have an agonist, you want to stimulate it, the last thing you want to do is to kill the cell that you're targeting. So that's why you want to knock it out. If you don't want the FC function, so why would you use an FC? FC has several functions. One function is, first of all, it makes the molecule much bigger. So all molecules, all proteins that are less than, let's say, 60 to 80 kilodalton, they are very quickly cleared by the kidneys. Very quickly means hours. So inject it and it literally goes down the drain. So an FC fusion already makes your molecule bigger, and it doesn't get cleared in the kidneys. So that's number one. Number two, antibodies, they are rescued from degradation in the liver and in endothelial cells. How does that happen? That happens by what is called the Bramwell receptor or the FcRn receptor. So when they are engulfed by pinocytosis, for example, in the endosomes, the pH is lowering. At a lower pH, it binds to the FcRn. The FcRn is recycled, gets back to a neutral pH, the binding is released, and the antibody is rescued from degradation. That's what I want to illustrate over here. FcRn interaction. We know the FcRn interaction, we modeled it, and we can manipulate it. So we can have mutations that increase the FcRn interaction and mutations that decrease the FcRn interaction. So we can actually play with the half-life of these Fc fusions. One example of playing with it is making it extremely strong. So one of them I mentioned over here. So Vivcard from Argenix is a Belgian company, and what they did was they just make the Fc with an incredibly high affinity for the FcRn. So it binds the FcRn, it doesn't come off anymore. That means that there's no more FcRn available in order to rescue all the other antibodies. So all antibodies become with a short half-life, so they will be cleared more rapidly from the body. Why is that beneficial? Well, if you have autoimmune diseases, you have antibodies in your body with a half-life of 3 weeks. That's a normal half-life of an antibody, of an IgG1. So and they are against your own antigens, so you want to get rid of them. So blocking the FcRn gets cleared out all the autoantibodies, and it really works in the clinic. So just an example. So we can play around with the half-life over there. Other mutations are also mentioned here. So some applications, for example, they benefit from higher concentrations. So you can give subcutaneous bolus injections. It's more practical than an infuse. But then you have to concentrate your antibody up to 200 mg per mil, and not every antibody can do that. Again, there are Fc mutations that we can try and in order to get that, together with mutations in the V domains, etc. But there are engineering technologies in order to get to that goal. So we can manipulate the activity of the Fc, we can manipulate the half-life of the Fc, and we can manipulate even the higher concentration, all by just working on the Fc. Right, that's about antibodies. Now, remember, 150 billion market only for the top 20 from the antibodies. So how are we doing with the bispecifics? That's on this table. And then you see that it's not really comparable. So most of the, and I'm talking about approved antibodies that are in the market, that are sold, antibodies that are still in clinical trials. They don't make money, they cost money because you don't pay for them. You're registered register in a clinical trial, and then you get it from the companies. So here you see that the total number of even more than the top 20 there, it goes to what was it, nearly 10 billion. So 10 billion against the 150 that we see with the top 20 of the antibodies. It's not a lot. But then on the other hand, look at the time when they were approved. They were all very recently approved, and the revenues that they make are not so much because they only are just becoming available into the clinic. And then look at the last column, that's the growth, the annual growth that we saw in the last years, which is for some of these antibodies really spectacular. So that's normal because they are just introduced, they are new, and they grow from zero to a lot. So main message here is like the bispecific antibodies, they're not an academic project anymore. I mean, an academic project including clinical trials. Clinical trials is also done in university hospitals, other hospitals, but they are done as research. Actually, clinical trials are research projects. So these are really market introduced, and they make a difference, or they're gonna make a difference into the therapeutic market, bispecific antibodies. And one more thing I want to point out is in what field. So I colored all the T-cell engagers. So one way of using bispecific antibodies is by having a T-cell antigen, so the CD3 activator, the CD3 receptor, and then cross-link it with a tumor antigen. So in that way you kind of like force the interaction of the T-cell with the target cell. So that's been, research has been going on for a long time. And all of these molecules are in green. So that's quite the majority of the applications. Other applications are in red, and that has to do with combinations of immune checkpoints. So these are the two fields that are emerging. So that's where the field is most probably heading in the coming years. A little bit more about bispecific antibodies, because the first thing is you need to make them bispecific, and there are several ways of doing that. What I try to highlight over here is some of the early solutions, protein engineering solutions, using fragments. And one of the first models here was by a company called Micromet, a small German company, was then later on bought by Amgen, and they call it the bispecific T-cell engager, and it's basically just two single chain molecules, which are the VL and the VH with a long linker, and then they are coupled together with a shorter linker. Here you see a model of this. Usually you draw it on paper like that, and here you see, if you model them, you clearly see that there's one VH-VL pair here and with a VH-VL pair there. In red you see the CDR3s, so that's where the antigen binding site is. So you clearly see these antigen binding sites, they are oriented out to the outside, so it's perfectly possible to use these molecules in order to cross-link two antigens. And another conclusion from this model is that this cross-linking will be done in a quite short distance, so shorter than when using a complete antibody with Fab arms that have the span of width like over there. Another solution was with macrogenics, which created the DART molecules, the dual affinity retargeting antibodies. So if you draw them on paper, they kind of look like completely different, but if you model them, you get exactly the same model, except excuse me, except with much less linkers and shorter linkers. But in the end, you get the same thing. The V domains, they orient themselves in very close together and with the binding sites towards the outside. So this DART model over here is kind of based on what we call a diabody, and you can make diabodies in several ways, single chain diabodies or assembled diabodies. So it's basically a diabody model. Diabody models together with single chain Fe models, so like the BITE models. So one thing that I want to show you here with these diagrams is that they are very close together. They're kind of rigid. They're not really flexible, and they don't have a wide span of range. So they're perfect to bring two antigens together. There's another model that has been used in several models. It's called the DVD. It was introduced by AbbVie, and it's just like two variable domains that you put in sequential order, and that is the model over here. Hope it's a little bit clear. So the first one is in the bluish colors, and the second one is in the like orange-like colors. And here you see that the CDR trees, they are oriented towards the same side, so you can expect a little bit of steric hindrance. If one binds, then most probably the other one will not bind anymore, and that can have its disadvantages or its advantages. I think I have a slide on that. I will come back to that later. One more thing. So a lot of development on the bispecific antibodies was using one of these models over here. So starting at the left, you had these models that were all based on a combination of these fragments that we just discussed. So either the BITEs or the DARTs, or just VHHs, which are single domain variable regions. Now these are very fast clearing antibodies. Like I told you, so they have a kidney elimination, so they are smaller than the 60 kilodaltons, so they immediately go out of the kidney. And that's it here, half-life less than a day, and in reality it's like 3 or 4 hours, and then they are eliminated. So are they valid? Are they clinically interesting? Yes, they are, and they have been used. Like we all know blinatumomab, Blincito, which is in the clinic, was one of the first that was approved. But it has to be given in cycles of continuous infusion. So you need to be in the hospital a whole week with an infusion in your arm, which is theoretically perfectly possible, but in practice, the medical institutions and the social security, so medical insurance, they don't like it because it's expensive. Also the patients, they don't like it because for every treatment cycle, they have to go and be hospitalized for a complete week. So it's something that is not too favorable. Also in theory, with a half-life of a couple of hours, everything that you pump in will, after 3, 4 hours, it's literally down the drain, so you lose a lot of your antibody. Then there's another class of molecules over here, which are called intermediate fast clearing, and they have a molecular weight which is above the kidney clearance, so they have a little bit of a longer half-life, meaning that the half-life there is between 3, 5, some of them 7 days. It depends on which study, and there are some examples over here. Some of them are even in the clinic, like the IMTAC, and there is one tribody also in clinical trials over here. Dock and lock as well, but that is, I'm sorry, but more for diagnostic purposes. What can you do then? So a lot of companies are still using these fragments, and they just add a serum albumin binder. So there's another protein, so you have the antibodies via the FC, and there's also human serum albumin, serum albumin, who also binds to the FcRn receptor and is also rescued. So the idea is there, if we have a binder to the serum albumin, we kind of piggyback right onto the serum albumin. The binder has to be pH independent, so it must stay on, and then we take advantage of the longer half-life of serum albumin. So you see here, a lot of companies have done that, and a lot of companies have made molecules like that, and they were tested in the clinic, and the half-life was found between 5 and 10 days. which is good. It's an improvement, but not a tremendous improvement from just avoiding kidney clearance. And if you compare it to monoclonal antibodies, so monoclonal antibodies have an average half-life of 3 weeks. So it depends on the variable domains. It depends on whether or not you make strange recombinant combinations there, so that can affect it. So most of the dosing frequencies of the monoclonal antibodies or derivatives is every 2 to 4 weeks. So here the Q stands for CADA, and the 2 to 4, and the W is weeks, and the D here is for days. So with the human serum albumin binder, it has an effect, certainly, but it doesn't come near the range of half-lives that we can see with normal FC bearing antibodies. So, knowing that, what did the companies do? So we already talked about the BITE, for example. So that was done with Amgen. So what they did is because they were in clinical trials, in phase 1 clinical trials with a lot of BITEs, and they are here in the last row over here. These were all the molecules that went into phase 1. One of these molecules, blincitomab over here, was approved and is now still in the clinic in a continuous infusion. So it is used and it is possible, but what they did later was they took that BITE structure and they grafted it onto a FC region, and they call that the BITE HLA, and the HLA stands for half-life extended. So they have a half-life extended BITE molecule, and that seemed to be very successful. So they were able to make it. They had their own model, and the star here in the schematics means that it's a knocked out, so it's a silent FC, because the activity of the molecule comes from the bispecific molecule. You don't need any ADCC or phagocytosis inducing activity from the FC. So you make a silent FC over here, but you do keep the half-life extension functions of the same FC. So here are some examples of this kind of molecules, so the HLA that are now in clinical phase. I told you about the diabody based models from Macrogenics, and they did exactly the same thing. So they were not so successful so they are a little bit behind on their clinical development, but basically they go into the same way. So they took their small dart-like molecules, they grafted it on to a silent FC in order to get the half-life, and then they get this. You notice that they don't do it in a normal FC, so they do it in an FC that is bispecific, that is meant by this symbol over here in the schematics, because you only want to make one-arm molecules. Why do you want to make one-arm molecules and not two-arm molecules? You can make two-arm molecules, but the active part here is an anti-tumor antigen, anti-CD3. And we also know that if you have a bivalent anti-CD3, you're gonna hurt the patients, because you're gonna stimulate T cells which are in the periphery, which are not in the tumor, but everywhere in the periphery. They will all start to secrete cytokines, especially IL-2. You will get high fevers, you will get a lot of inflammatory cytokines, and that is a phenomenon called cytokine storms, and that can kill people, and it's not beneficial. You just get sick or you can die, but it will not cure your cancer. So you have to activate the T cells only at the tumor and not in the periphery. That's why they keep the CD3 binding monovalent, and it must be strictly monovalent. You could use the other arm for example, adding other binders to the tumor cell, etc. And that is developments that are happening, but it's in order to get the monovalency of the CD3 that we use bispecific FC regions. Macrogenic here does it by engineering the interface, and here, this is the previous slide, so Amgen is doing it by, and this is kind of strange to see, they make a one big single chain FC, Is an enormously long linker over there. So they prefer that, and it seems to work in their hands. Coming back to these DVDs. One of the models that are still in development is something like this, and because they want to go for bispecifics, not immediately for T-cell engagers, but for other bispecifics like immobilizing or neutralizing cytokines, etc. And they wanted to do a bispecific bivalent by coupling these FAB regions. Now, if you want to do that, it's not if you want to do that, it is happening. Some of these molecules are in clinical trials, but then you have to work with these common light chains. So you have to get libraries, you have to prepare your libraries that your antibodies are compatible with these common light chains. So Chugai is developing it, they call it LinkIG. Epimap is a Chinese company, and they call it FABs in tandem, so fit IgG, and that's this model. This model is probably what inspired AbbVie to say, like, okay, why use these FABs? I just gonna graft the second V domains, the ones from here, and graft them immediately onto the first V domains, and I call that dual variable domains, so the DVD IgGs. I mentioned that a few slides ago. This is again the model. So here you have the first pair of VHVL. Here's the CD3, and here is the second pair, and here's the CD3 there. I already told you that this is a special model because it gives you the impression that there will be some conformational hindrance. If one antibody binds, or one antigen binds, then the other one will most probably be blocked. Now, in all the applications here, except for this one, it doesn't really matter. So you have one antibody which can either bind and neutralize one factor and the other factor as well. So it's not necessary that both of these factors are bound by the same antibody. It's just like you have one molecule and you can neutralize two different soluble factors. That's what AbbVie is mainly aiming at. Now, Sanofi is also using that technology, and they have kind of like a spectacular, for me it was a spectacular event. So they did created T-cell engagers with that, and on one arm, this arm here, they had a normal antibody targeting the tumor antigen, and on the other arm they put a DVD which it was targeting CD3 and CD28. And that was something like, wow, you must be crazy to do that. Why? Some people here have worked with T cells, and if you work with T cells, you activate the T cells. How do you activate the T cells? You put beads in it which are coupled with CD3 and CD28. And then you see that these T cells, they go bananas, they start dividing, they start secreting all kinds of inflammatory cytokines. And that is exactly what you don't want to do in the periphery of a patient. That's absolutely not. You want to do that at the tumor site, but not in the periphery. Still, these antibodies from Sanofi, who have a CD3 and a CD28, they are in the clinic and they are fairly, they passed already the phase 1 toxicity studies, and they're good to go. So, but if you look at the molecular prediction over here, then you can see that if, again, I already told you, so if this variable domain pair is binding an antigen, then most probably it will inhibit the binding of the others. So this antibody is a trispecific antibody, but it either binds, so it binds to the tumor, and then it either binds the CD3 or it binds the CD28, but it will never cross-link CD3 with CD28. So the CD3 and the CD28, they will come together when there is enough of this antibody accumulating at the tumor site, giving this specific CD3-CD28 couple at the tumor site and not in the periphery. And that's probably why it works, which is great. It's like a big improvement. Right, we talked about bispecific antibodies, and we talked about the fragments again, and you've seen this slide before. So in normal antibodies also, we can make it silent, we can reduce viscosity, we can increase the half-life. All of these positions are known, we can manipulate them in order to get these bispecific antibodies. But then with a bispecific FC, we need one more step of manipulation, and that is we need to engineer the interface over here. So you see that this is the hinge and this is the CH2 and here is the CH3. So it's the CH3 that is binding and that is keeping the FC together. So it's that interface between the two CH3s that we can manipulate in order to make it bispecific, and as bispecific as efficient as possible, because otherwise you get homodimers, you have to, you complicate the downstream processing, just like the epicuzumab that we discussed before. How can we do that? There's a lot of efficient technologies there. The most efficient, or the most popular at least, is known like knob into holes. So that's the Genentech people again who did that. So you go to one of these CH3s and you see where when they interact, they have a hydrophobic core which is interacting, which is surrounded by charge interactions, by charge bridges. So what they do is they went to the hydrophobic core, said like, okay, this amino acid is interacting with that amino acid. I'm gonna make this one, which is a small one, and I'm gonna make it bulky. I'm gonna make it, tryptophan is one of the most bulky hydrophobic amino acids. So they put the tryptophan there. Of course, then you create a bulk, which they call the knob, and that's useless. But then they went to the other side and they said like, okay, here there's a couple of big amino acids, I'm gonna make them small, and they changed them to serines or tyrosine, small amino acids. So they created a hole. That's why they call the technology a knob into hole. Works quite good. 90% of the products that you make are bispecific, are heterodimerization. So you only have to remove like 10%. Another way of doing it, I told you, so you have the hydrophobic core and around it there is some charge bridges. So that's the Amgen way of doing it. So they reversed the charge bridges. So it feels positive, positive here, negative. They put all the positives in one chain and all the negative ones in another chain. So they reversed the charge bridges, making that only the heterodimer is stably formed. That's one advantage. The second advantage is you are changing the PI, because you have to purify it later. And if you purify it later and you use an ion exchange, you do it in a gradient, you will see that the homodimers and the heterodimers, that they elute at different positions. So it's also very easy. So not only it favors the heterodimerization, but it also makes the downstream processing easier. So because in the end you need to purify 100% pure heterodimeric antibody. On the slides there are some other engineering technologies. Most of them were like completely engineering the interface. GenMap, for example, has a technology where they use IgG4s. They have a tendency to form heterodimers. IgG4s in your body, they make spontaneous bispecific antibodies at random, so they fall apart and then they assemble again. You can stabilize that interaction by changing the hinge a little bit, and then you can do the same thing but then more stabilized by a mild reduction and a reoxidation. So that's the GenMap way, but then afterwards it's not never 100%, so you have to purify the rest away anyways. Also re-engineering the complete interface. There's several solutions published, but most of them were kind of like an invent round. Remember that the knob in hole engineering was done by Genentech, which is the Roche group. They filed patents on it, they got the patents on it, so you could not use it without paying a huge license fee to Roche or to Genentech. So in the meantime, those patents have expired. Knob into holes is free to operate, and actually everybody can use it, and now everybody is going that way. So most of the FC containing bispecific antibodies actually use this knob into hole technology, and not only from Roche, from all kinds of companies, competing companies as well. Right, that's how we get a heterodimerized FC. Now, we get the next problem. So if you combine that with a fragments, or at least one arm of them has fragments, then you don't have any problems. If you want to make a IgG-like bispecific antibody, you have two light chains, and there's your problem. So here's what can happen. So this is the correct molecule, but you can have one or both of these arms where the light chain is mispaired, because a light chain can pair to any FD chain. So you get some kind of a random, sometimes there's some preference for the binder, but you get a random light chain interaction, which is hard to control because they all look the same. They look the same on a gel, they look the same on a column. You need mass spec analysis in order to really go and tell them like, okay, no, this is they will still bind, because sometimes the heavy chain still has some binding affinity, so you will still have some affinity. So it's kind of like a difficult problem. So another problem that we need to solve. I already mentioned the common light chain, but the common light chain, so that's a kind of like a dummy light chain that is there, that is not binding anything, but it's just supporting the heavy chain, and then you can use that. But you have to build that into your libraries, and so you're restricted to these libraries in order to get your binders. Now, what can be done? The same people that did the knob into holes, they came up with a very elegant solution called cross-map. So what they did was they took one light chain, and instead of the CL, so the light chain is a VL and a constant light, so they removed the constant light and they put the CH1, so the CH1 from the heavy chain instead of it. And then on the other side they had the VH coupled to the CL. So they just crossed the constant domains of the fab. It's called the cross map. And some of the models over here, which are in the clinic, so you see here that most of them are doing quite well. There's one here, like Profit, yeah. It depends on the application, of course, but antibodies are really in milligrams that you give them. T-cell engagers are given in micrograms. So the blinatumomab is dosed in 15 micrograms per dose. In some clinical trials for T-cell engagers to tumor antigens, you reach therapeutic doses or maximum tolerable doses at 50-60 microgram per body weight. So let's say that an average person between men and women alike is 60 kilograms, so that's 10 micrograms per kg. While if you go from antibodies, you need like 1,000-fold. You need milligrams, tens to even 100 milligrams. Okay. Where was I? Yes. So here, like this model over here, that's this one. Glofitamab. It's a double CD20 for hematological tumors with a CD3. If you're interested, follow it. The clinical results there are very, very encouraging. So this seems like a really nice model. Half-life is quite good, and now we can make truly IgG-like bispecific antibodies. That's the cross-map technology. There are other ones. Lonza, for example, they offer what they call a bilock. They just moved the cysteine bridge a little bit. Chugai is offering what they call a fast Ig. That's also charge reversal, but then in the fab. Wuxibody, they use the constant domains of a T-cell receptor, and then you have the Samsung. Samsung not only makes mobiles, but there is a Samsung Biologics as well. So they also have a different. What they did is they took the knob into holes CH3 and they inserted it into one of these fabs. Okay, these are all again for me. my interpretation is these are inventor rounds because cross-map technology is not free to operate yet. It's a Genentech technology. So these companies, they offer kind of like an alternative, which is also not free. A Biloc, you pay 150 just to start, and then when you enter clinical phase, it's like 1 million, something like that. But that's confidential. No, we had that one. Go. Okay. Is that the end of the story? No, that's not the end of the story. The end of the story, what I show you here is, you've probably already seen lots of variations of that. This one is called the periodic table of all the bispecific antibodies, and you can expand that, or your fantasy is just a limit. You can add single chains, diabodies, single domains to whatever site that you want, and make all the combinations that you want. It's theoretically possible, and most probably, if you want to do it, if you want to produce these molecules, you will probably be able to do it. But is that a solution? It's not a solution to anything. The solution is looking at the function. So the function, for example, here we come back to the T-cell engagers. So the T-cell engagers, they need to mimic the function of a normal MHC T-cell receptor interaction. Now, when a T-cell interacts with a target cell, it forms what is called a synapse. And then in that synapse, there are also co-accessory molecules, which are bigger, and they are driven outside of that synapse. And then the real interaction, so this interaction here has to happen in the center of the synapse, and that center of the synapse needs to be very, very close together. So they need to bring the membranes very close together. So you can immediately imagine that some of these models that we have discussed in the previous slide, that they're kind of bulky, and they're a bit too big because the span of weight is too large. Coming back here to the BITEs, so this is a model from Amgen, from the BITEs. So here you see a BITE with a tumor antigen and the anti-CD3 compared in a synapse with the normal interaction, so the T-cell receptor with the MHC, and now expanded with the FC. But that is probably how it enters into this synapse. So the FC is here is just to extend the half-life, and then the interaction over here in that synapse is functional. Myself, I was involved in optimizations of such molecules where we compared different technologies, like different ways of making diabodies, of making these BITEs, with different linkers, etc. And you get different outcomes. So for example, you can screen for a whole panel of cytokines, so which cytokines are when produced. Remember, there's also a difference in these T-cell engagers. Cytokine excretion is not immediately correlated with target cell killing or toxicity. So there are ways, there are conditions where you can have a more efficient or just as efficient way of killing the target cell without the secretion of a lot of cytokines. And that's actually what the clinicians are looking. So they like that. So kill the target cell, but not have too much cytokines, because the cytokines you need to control. Okay. Right. Bispecific antibodies. Where can we go? Where is the field going? I have a few more minutes in order to do that. So I showed you this table. I showed you a lot of them are these checkpoints inhibitors, and pembrolizumab, for example, is a large field. It's 25 billion a year on its own. Pembrolizumab, all the PD-L1, all the PD-1, the LAG-3s, etc., they don't work alone. They work in combinations. We already know that there's different antibodies. If you combine them, then you get a better results. So this might be really a field for bispecific antibodies that is only just starting, because we're only starting to realize what of these immune checkpoints are important, when are they important, what will be the effect of combining, of a combo of binding, and can we do that in a single molecule. So that is one way of doing it. Few examples here. So here PD-L1 antibodies alone, so the ones that are here that are I divided it in regions. So in China, preclinical, phase 1, phase 2, phase 3 and approved. So this is only PD-L1, but then you see that around, let's say, 120, and about half of that number, so in addition to those, are combinations with something, combinations with other inhibitors, combinations with co-stimulatory molecules, or combinations with tumor targeting molecules. So that's clearly a field that is being exploited. The same thing for LAG3, the same thing for PD-1, but even PD-1 is even more advanced. There's even more going on. Just if you think of, like, I'm going to develop another PD-1 or another PD-1 bispecific, just give you these numbers over here. So this is the number of trials of these antibodies which are already approved, that already have been done. So that's the disadvantage that you have if you enter the market now for a PD-L1 antibody. So you probably don't want to do that. So we're not going to do anything with PD-1 anymore. No. Improve it, make it better, not do the same thing again with another antibody or another binder. That's not going to work. Nobody's going to invest in that. What happened here is a Chinese company, and they added VEGF to it because they know that when you give these two antibodies separately, that you get an increased effect, and when they combined it into the same molecule, they got a better effect. They showed that in preclinical trials, and they showed that in clinical trials. So this curve over here is a clinical trial, and for the first time they saw a checkpoint inhibitor really outperforming what was going on. So everybody went bananas, everybody went crazy, and everybody wanted to do that. So first of all, probably the mechanism is that by binding VEGF, because VEGF is a dimer, you start to cross-link the compounds, the bispecific antibodies, and you get a multivalent antibody. You don't get it in the periphery because most of the VEGF is concentrated around the tumor site. So you get a multivalent binder around the tumor site, getting a better effect on the tumors. So that's how people believe right now that it works. And it is what happened. So this is an example, especially in China, especially Chinese companies. So everybody was like, don't make it too complicated. So what you want to do is you want VEGF, you want PD-1, or you want PD-L1. What do we have? We have Avastin. It's free to operate. It's already free to operate. That's what this circle over here says. So we just glue a PD-1 or a PD-L1 binder to it, and we have a new product, and we test it. And given the results of this summit, you see here, these are the numbers of the deals. So they got 200 million just for the rights. Here, 600 million just for the rights. 1.2 billion just for the rights. Just all based on this one clinical trial that says, like, okay, if you combine it with VEGF, it's better. So yes, you can still do things in this field, which everybody knows. And the other way, the same. So the first slide was based on Avastin, and we glue a PD-1, PD-L1 binder to it. And this is the opposite. So you start with known PD-1 or PD-L1 binders, which are not yet free to operate, but they will come off patent, this one next year and this one in a couple of years. So who cares? By the time that we are in the clinic, they are free to operate as well. So don't make life too complicated. Just glue them together, do the clinical trials, see that you find a sponsor, and go to the clinic, because it all happens in the clinic. Last word, a few new directions in bispecific antibodies. Where are we going? So one of the things that is rapidly moving is biparatopic antibodies, and I had the pleasure to work with Claudia some years ago on that, and we proved that even trispecifics, that they induce internalization into the cell and then rapid degradation. And this is a principle that now has been brought to the clinic for HER2, but also for other antibodies over here, and it works quite well. Especially, for example, if you combine it with ADCs, so where you put a chemotoxic drug coupled to the antibody, because it's internalized and then it goes to the lysosomes and it's degraded. So when it's degraded and all the toxic stuff comes off, goes into the cells and kills the cells. So you can do it with ADCs and you can do it with radiotherapy. Another thing that I had the privilege to work on is with targeted membrane protein degradation. So basically what you do is you don't want to block a receptor, you want to degrade the receptor, and that has its advantages. And you can do that because you use the ubiquitin system, and in the ubiquitin you have a ligase, and some of these ligases are also membrane proteins, so you cross-link ubiquitin ligase to your target membrane, and then on the inside of the cell it will get ubiquitinated, gets to the lysosome and gets degraded. So that's a different mechanism than just blocking the antibody. That's number one. And number two is there is a whole range, there's more like 100 of ubiquitin ligases with very specific cell type or tissue type expression. So if you know that your tumor is hot for one particular ubiquitin ligase, but for example normal tissue is not, then you pick that particular ubiquitin ligase and you spare the normal tissue while still degrading on the tumor. Last slide. Another interesting development over here. There's the masked antibodies. So in order to get more specificity, because we know how to get the toxicity there, is more like the specificity. We only will have this killing at the tumor site and not at the normal tissue. You can mask these antibodies. There's several techniques. There's some examples here on the slide, but basically you make sure that there is some moiety in the antibody masking the binding, inhibiting the binding, and you make it protease sensitive. And you know that in tumors, around tumors, there's a lot of metalloproteases, other proteases which go around because tumors, they need to degrade the extracellular matrix in order to be able to invade. So and you make them cut away this inhibiting part. That's the masked one, so higher specificity. Great, and that's the end of my presentation. Just in time, I think. So the conclusions: bispecific antibodies, they are taking their place. The technology is matured, it's available, most of it is or is becoming free to operate. Form and function, very important, not just like, well, we can do everything and we can make all kinds of variations. These variations should be to make a better make it better fit to the biology of what the antibody is have to do. We know the toxicities, we know the PK, the PD. They are not hurdles, they are manageable. Nobody is afraid of them anymore, even not cytokine storms. We can handle them in the clinic, so there's no medical doctor who will say like, no, I will not give that to my patients because of the cytokine storms. They know how to treat it, they know how to handle it, it's understood. The CMC, so the production issues, it really needs to be bispecific and 100% pure. It's understood, it's under control, we know how to do it. And it has a major impact in T-cell recruitment, so as we've seen, and in the immuno-oncology field, with new applications on the horizon. Thank you.
Thank you, Nico, for this overview in this hot and fascinating field that has become really, I think, the future of antibody field. Questions? Lucio, Pastore, Professor Pastore.
I was just wondering, since you mentioned also the problems with purification of some of the antibodies, if for the smaller one, would be an option to use RNA to administer them as in RNA form, aside from post-translational modification, and obviously you had to check whatever it happens where you administer them. But do you think it's a viable idea? Could it work? Could it be cheaper than purifying antibodies?
Okay. I think it certainly is a valuable idea, and I think every valuable idea should be worked out. But probably this technology needs some optimization in order to get to where we are. Would it be cheaper? Don't go that way. Don't go there. Once these antibodies go into production and you have the advantage of scale. I was discussing with people from Amgen, from Genentech, and they say like at the levels that we make, for example, Herceptin or rituximab, we make them, the costs of making 1 gram of antibody is what a patient probably needs from a normal antibody. 1 gram, you can make it for $100. So the cost is not an issue. If you can do the RNA and it can produce it and etc. So then it of course it's viable. So don't stop researching these ideas. But the cost on its own, I would not say that.
My concern would be the post-translational modification, of course, because when you produce it, you obviously optimize your production of the antibodies with what you want.
Yes.
When you give the RNA, you have to make sure that the RNA is modified the same way.
Yes, but also the RNA, you have to produce it. You need the downstream processing, etc. So yeah, exactly. No, it's certainly do work out that idea, but at the moment, I think one of the first slides is where are bispecific antibodies right now in the market. And actually, the point I wanted to make is like if you compare it to antibody market, which is matured, is still very small.
What I feel that is probably you get more investment if you just make the antibodies right now, because there's clearly let's say an excitement about this field, while the RNA is still
Exactly. So if you want to get investment and you want to go, so like, okay, I have something and I'm pretty sure, look, here are the data and I can get it into the market. Can you give me 20 million? Because that's basically what you're asking. So, and then you say like, yeah, but it's an unknown technology and RNA, etc. Then they back off. They said, no, you work out the technology first and then come back. So, because this technology, it works. They don't doubt it works. It worked before. One of the questions, because I'm involved in creating companies, etc. So one of the questions we always get from the investors is, for example, if say knob into holes, how many antibodies are in the market that use the knob into holes? So here's the list. It's like 15 antibodies there. Oh yeah, okay, that's fine. And then they are. So if you say none, but it will work better, but none. It's my 20 million. you know.
Other questions? I have one also for the students. Can you give us some clinical response elements, average, and what are the best formats among those you have shown, also in terms of clinical response?
Well, glofitamab was one that really I pointed it out. Where is it? That one is really good. Doing this one over here. That's a CD20, CD3 from Roche, and so the advantage there. So you probably noticed in that list, a lot of them they're monovalent, targeting the tumor. And obviously also monovalent for the T cell. So the monovalent for the T cell, that is very important. You can't change that. But multivalent targeting, we're just having that discussion. So me personally, I think that instead of affinity targeting, we should consider avidity targeting. So target with a lower affinity towards the tumor site and get an avidity targeting, because you most probably you will have a tumor-associated antigen that is not 100% unique for the tumor. So it's enriched in the tumor, but there's also other normal tissue which has it. If you kill that other tissue, you run into problems. That's one point. So getting more to the avidity, like clofitimab is doing, for example, and you can expand on that idea. Other people are also working on the CD3. The same thing. So higher CD3 probably gives you a better activation of the T cells, but also meaning higher cytokine induction. So a lot of people are modifying the CD3 affinity to get a lower T cell activation, but then combined with a lower half-life and a longer treatment time, they get the same effects, but with less side effects. So that's another way of looking at it. But that's where the field is moving.
But with respect to blinatumomab, that is about 70% overall response, right? With the first bite.
Yeah.
Is it this the response rate?
No, it depends on the tumors. Blinatumomab is a hematological tumor.
Yeah.
And that is also clofitimab is CD20, so it's also for lymphomas. So lymphomas, most of the successes of the T-cell engaging bispecific antibodies is with lymphomas or myeloma, so with hematological tumors. Solid tumors is
still a challenge.
Is a big challenge, and there is the checkpoint inhibitors, etc. That I don't think that any company is now starting any clinical trial anymore without a combination of a checkpoint inhibitor. So if they have a T-cell engager for a solid tumor, they will probably do it alone because they need some toxicity data in the phase 1, but they will immediately start it combined with some checkpoint inhibitors. That's why they're selling so good, because everybody wants to combine their product with a checkpoint inhibitor.
Other questions? Also from home, someone? Okay, I would speak for hours of this topic, but I think we are now in time for lunch. There is a question.
Hello, Professor. Thank you for the seminar. And I just have a question that what are the challenges for translating the biospecific antibodies from bench lab to clinic, lab to market?
Yeah. First of all, don't make it too complicated. Second of all, what you probably are focused on in academia, or at least when I was in academia, I wanted to understand the mechanisms, etc. That is important. And when you file for a clinical trial, you need to fill in what is called the common document in order to ask for permission to start a clinical trial. And that's important that you have all the data there to also convince investors to give money in order to do that. The mechanism is certainly one point, but what is just as important, or maybe even important, is preclinical. And preclinical is not the same as the mechanism of action. So mechanism of action is I can explain what happens, but most probably everybody who is there with the money, there's like, yeah, that's one way, but it's probably doing something else as well, because that you say that the marker is on those cells, but it's also on other cells, and what is doing there, so you never completely understand it. What is important is what is the effect, and how can I show the effect, and what is just as important is what is the toxicity, and how can I have some tox models, preclinical tox models that can demonstrate that. So these data are very important, and if you combine these data, so the effect and a real effect in most probably in vivo, so in vivo meaning in an animal model, in mice, and then also a tox model that goes with that, and then you're good to go.
Thank you.
Okay, so if no other questions, thank you, Nico, again. And if you want to ask him other questions, we are around. Bye.
Okay.